Today we are going to discuss about the blood. The study of blood and lymph is called as hematology. Hematology is nothing but the study of blood and lymph is called as hematology. Blood along with lymph. Now majorly blood is a fluid connective tissue. Blood is a fluid connective tissue, that means which connects through all parts of the body. And we have two types of bloods, which is called as arterial blood and venous blood. Coming to the color of the blood, the arterial blood is bright red in color and the venous blood is dark in color, which is also called as brownish red, brown in color. And this is bright red in color because of the presence of oxygen, and because of the absence of oxygen it is in brown in color. And arterial blood carry pure blood, hence it is called as oxygenated blood and it is bright red in color. And coming to the quantity, how much of amount of blood each human being having, we have 5 liters of blood approximately. Every individual have 5 liters of blood approximately. And coming to the functions, what are the functions of the blood? Mainly the blood will be useful for the transportation of gases, mainly transportation of oxygen and carbon dioxide, and also remaining gases can be transported with the help of blood. And coming to the excretory function, the blood also performs the excretory function. It can excrete the waste products which are not useful for our body through the process of excretion. And we also come to the nutritional function of the blood. What is the nutritional function of the blood? The nutrition which is used for our body, for example glucose, amino acids, lipids and proteins which are useful for our body will be produced and stored in the blood. And coming to the fifth function, acid base balance. This is maintained by the blood because of the maintenance of the acid base balance. We know acid means the pH value will be less than 7, and if pH is more than 7 we call it as base. And our blood will maintain the balance of acid base balance, which is also called as buffer system. The acid and base should be in equal quantity so that the pH of the blood should be maintained. And the pH of the blood is 7.4, that is not acidic or not basic, it is neutral, that is 7.4 is neutral. And the large amount of acids are produced daily because of the metabolism. We know that our stomach produces large amount of HCl, which is used for the digestion of the food products, and the blood has the capacity to maintain this acidity as well as to neutralize. It will maintain the neutrality, and which is also called as homeostasis, which is called as maintenance of the neutrality. And it also useful for the transportation of the regulation of temperature, which is also called as temperature maintenance, which is also called as homeostasis. And it is also responsible for the transportation of hormones. Transportation of hormones is also done by the blood. And last but not the least, we can also see the function of defense mechanism. The blood will be helpful for the defense mechanism. How it will be in defense mechanism? It will be producing the antibodies against the antigen, and it will also help in the transportation of the antibodies produced in our body. Just like even with the help of production WBC, the white blood corpuscles, which are also called as leukocytes, with the help of these leukocytes the blood will be acting as a defensive, and it also kills the organisms like bacteria, virus and fungi, and helps in the defense mechanism. And we can even see one more function, main function, as water balance. See, the water should be in a equal quantity and in a balanced quantity in our body. If there is excess water, it will be releasing out with the help of the kidney, and if it is less amount, it will be helpful in storing of the water. Hence, the water balance will be maintained by the blood. And coming to the physical properties of blood, how the blood will be appearing, you can come to the physical properties. Coming to the physical properties, the pH is 7.4 as discussed earlier, and color is right bright red in color, and brown is for the venous blood, as we already discussed this point. And coming to the specific gravity, the specific gravity of the blood will be 1.035 to 1.065. And we can see the viscosity is nothing but the liquidity, and the presence of blood cells determines the viscosity. The viscosity is 3.5 to 5.5. This is the normal value, that is, this is nearly 3 to 4 times of that of water. This is 3.5 to 5.5, and this is 4 times more of that of water. Next, coming to the composition, blood is composed of blood cells and also plasma. We can see plasma. Blood cells constitute about 45%, and plasma constitutes about 55%. Remember this, blood is not only liquid, but it contains blood cells, 45% of blood cells and 55% of plasma. Now we can see what are the types of blood cells present in the blood. There are three types of blood cells mainly: RBC, which are also called as erythrocytes; WBC, which are also called as leukocytes; and platelets, which are also called as thrombocytes. These are the three types of cells present in our blood. And coming to the plasma, which accounts for about 55% of the blood, we can see the water is 91% in this plasma. 91% is water, and remaining 9% we can see solids as well as the liquids, solids as well as organic material. Solids will be 1%, and organic matter will be around 9%. Understood? The blood consists of 45% of these cells and 55% of plasma. In this plasma we can see water 91% and 9% solids and organic. Solids will be 1% and organic will be 9%. In this organic we can see, this is 8%. In this organic we can see sodium, potassium, chloride, magnesium, phosphorus. All these are the organic contents where we can see sodium, potassium, chloride, magnesium, phosphorus constitutes about 8% of the plasma. Okay. Now we come to the plasma proteins. See, the proteins include globulin and albumin. Plasma proteins are mainly constituents of the plasma, and the normal level is 6 to 8 grams per 100 ml of the blood. In 100 ml of blood we can see 6 to 8 grams of the proteins. This is the main important part. See, if in the blood, if proteins increases, this will be a problem for the man. Even if decrease also, it will be the problem for man. So if there is increase in the protein, we call it as protein urea in the urine. If the protein is more, we call it as proteinuria. In the blood, if the protein is more, we call it as proteinemia. Mia refers to blood. If in the urine protein is more, we call it as proteinuria. If in the blood, if protein is more, we call it as proteinemia. And coming to the types of proteins, we can see albumin, we can see globulin, and we can see fibrinogen. Albumin is 3.5 to 5 grams per 100 ml, and globulin is T2 to 3.5 per 100 ml, and fibrinogen is 0.2 to 0.3 grams per 100 ml. Remember once again, the albumin is 3.5 to 5 grams per 100 ml, and globulin is 2.2 to 3.5 grams per 100 ml. Albumin is 3.5 to 5 grams per 100 ml, and globulin is 2.2 to 3.5 grams per 100 ml, and fibrinogen is 0.2 to 0.3 grams per 100 ml. If albumin is more, we call it as albuminuria. If globulin is more in the blood, we call it as globulinuria. And fibrinogen is there, we call it as fibrinoemia. See, mia refers to blood, albuminemia, globinemia. Next, we can see application part of these knowledge of the blood cells. See, if in applications we can see the normal albumin and globulin ratio should be 1.5:2. You can see this, the albumin and globulin ratio should be 1.7:2. Okay, and if it is, this will be increased due to decreased synthesis of albumin. If they have any chronic liver diseases, the production of the albumin will be decreased. Because of this, albumin balance will not be maintained. In the condition of the chronic liver diseases, the production of albumin will be reduced. Because of this ratio cannot be maintained. In some other conditions like liver diseases and also autoimmune diseases also, this condition may occur. So the ratio of albumin and globulin in the blood should be around 1.7 to 2. And coming to fibrinogen, what is fibrinogen? Fibrinogen is important because it is synthesized in the liver and helps in the blood coagulation. Whenever the blood is coming out, whether it is damage to the blood vessel, blood should coagulate at a certain time, and which is called as clotting time. If blood is coagulating because of this presence of this protein called fibrinogen. And coming to the synthesis of plasma proteins, how these plasma proteins are synthesized? The plasma proteins are synthesized mainly in the liver. We can see the production of plasma proteins. Plasma proteins are synthesized in the liver. So, if the liver is damaged, these plasma proteins cannot be produced. So, liver has a major role in producing the plasma proteins. However, immunoglobulins are synthesized with the plasma cells. What are immunoglobulins? Immunoglobulins are the antibody particles which are generated in response to the antigens. These are the immunoglobulins which gives immunity to our body, and these immunoglobulins are produced by the plasma cells. Just remember, these immunoglobulins are produced by the plasma cells of the B lymphocytes. Plasma cells of the B lymphocytes. You can see plasma proteins can be separated by blood by 4 processes. You can see the 4 processes by which the plasma proteins can be separated. First is electrophoresis, second is precipitation, third is fractional precipitation, and the fourth one is sedimentation. All these are the methods used to separate the plasma proteins. Coming to electrophoresis, with the help of electrification or centrifugation, we will be separating the proteins. And with the help of precipitation, that is allowing the blood particles or the blood cells to decompose, to get down, is nothing but the precipitation. Fractional precipitation is allowing the different parts of different particles separately to precipitate, and sedimentation is allowing freely so that the particles will be separating by themselves. And coming to the main part, what is the function of the plasma proteins? We are discussing about proteins so much, and what is the main function? They help in coagulation. The main part is they help in coagulation of blood so that the blood bleeding time will be reduced. Coming to second one, they help in defense mechanism by producing the immunoglobulins. And third one, they are used for the transportation. And fourth one, they are used for the maintenance of the osmotic pressure. Maintenance of the osmotic pressure means they can able to maintain the pressure so that the albumins or globulins and plasma proteins can be in a required quantity, and they will allow only some substances to get inside and some substances only to go outside, which is called as osmotic pressure. And they also maintain the acid base balance as well as the functions of the blood we have discussed earlier. And they will maintain the viscosity of the blood. Viscosity of the blood is also maintained, and they also help in the formation of the Rolox formation. What is Rolox formation? This is important. If RBC are piling up of the RBC in the form of just like a coins is nothing but the Rolox formation. You can see the Rolox formation is piling up of the RBC one by one as like a coins is nothing but the Rolox formation. And we can see in applied physiology what we can see, hypoproteinemia. What is hypoproteinemia? This refers to the decrease in the presence of protein in the blood. Decrease amount of protein in the blood is called hypoproteinemia. See, mia refers to blood, protein is normal protein, hypo refers to less. The less amount of protein in the blood is called as hypoproteinemia. The normal, there will be so many reasons for this hypoproteinemia. Some of the reasons are hemorrhage. See, hemorrhage means bleeding. Bleeding may be one of the reason for decreased protein. And one more reason may be the burns may be the one of the reason. And some of the reasons like malnutrition, starvation and malabsorption. Malnutrition and starvation also may lead to the decrease of the proteins. And we can even see some of the disease conditions like some of the disease conditions like chronic liver disease conditions, we can able to see this proteinemia, that is hypoproteinemia. And even the renal disease, that is kidney disease, also may lead to the disease conditions called as hypoproteinemia. And we can see even this condition of hyperproteinemia also. What is hyperproteinemia? Because hyperproteinemia refers to the increase of the proteins level in the blood. The reasons for this dehydration, because of the loss of water in the body, hyperproteinemia occurs. And we can even see acute inflammatory conditions. Acute inflammatory conditions also will lead to the hyperproteinemia. And we can see even multiple myeloma. What is multiple myeloma? It is a disease condition of cancerous like condition, and even this condition proteins will be increased. And coming to the briefly description about the cells of the blood, we have discussed there are three types of cells: RBC, also called as erythrocytes; WBC, also called as leukocytes; and thrombocytes, which are also called as platelets. And these cells have their own importance in the blood, because RBC are important, WBC are also important, platelets are also important for maintaining the blood. Now coming to the hematopoiesis. What is hematopoiesis? Hematopoiesis. Poiesis refers to the production. Kindly remember, poiesis refers to the production, and hematopoiesis means production of blood is nothing but the hematopoiesis. How the blood will be produced? See, blood will be produced from the mainly red bone marrow. We have bone marrows, red bone marrow. Mainly the children is having more amount of red bone marrow, and we have yellow bone marrow also. But red bone marrow will have the maximum capacity of production of the blood cells. Blood cells, maybe RBC, maybe WBC, maybe platelets. but red bone marrow will be used for the production of the RBCs, WBCs and platelets. And we can see it at what stage the blood cells will be started producing. From the third week, you can see from the third week of gestation, from the third week of gestation to third month of gestation, the erythropoiesis will be taking place with the help of yolk sac. See, we have three layers of the fetus: amnion, chorion, yolk sac and allantois. In this, the yolk sac will be useful for the production of erythropoiesis, that is production of blood cells from the third week of gestation to third month of the gestation. From the third month of gestation to fifth month of gestation, we have liver. Liver and spleen will be producing the blood cells. Kindly remember, from the third week of gestation to third month of gestation, yolk sac will be functioning as the erythropoietic organ. From the third month to fifth month of gestation, that is intrauterine period, liver and spleen will be acting as a erythropoietic organs. And from the fifth month to the up to the time of delivery, red bone marrow will be the erythropoietic organ. We can see these three are the erythropoietic organ, and this is an important to remember. From third week, yolk sac; from third month, liver and spleen; and from the fifth month to up to delivery, we can see red bone marrow will be acting as a erythropoietic organs. Now coming to RBC, the importance of RBC. What is the importance of RBC? Red blood corpuscles, which are also called as red blood cells, they have circular disc-like structure and the nucleus is absent in the RBC, and their size is 7.5 millimicrons, and a red pigment called hemoglobin is present in the RBC because they are red in color. Because of the presence of hemoglobin, RBC are red in color, and the lifespan of RBC is 120 days. That is, after production, they will be living only for 120 days. And the normal shape is biconcave shape. The shape is biconcave shape, that is dumbbell shape. The shape of RBC is dumbbell shape, and the advantage of this shape is that they can able to enter. The central portion is thinner and periphery is thicker, and because of this shape they can able to enter into the capillaries also for the diffusion of the gases. Next, composition. In composition, we can see the RBC is composed of 60 to 63% of water and 37 to 40% of solids. As we have seen in plasma, we have seen 91% of water and 9% of solids as well as the organic solids. In the same way, in RBC we can see 63% of water and 37% of solids. The solids include the same potassium, enzymes, ATP, phospholipids, cholesterol, etc. All these are the solids which are present in the RBC. And coming to the structure, the RBC has layers of lipids, phospholipids and lipids, L, PL, L. This is the some of the structure. The structure of RBC contains lipids, phospholipids as well as lipids, and some of the glycolipids also. Glycolipids. The lipids which are containing glucose are called as glycolipids. And we can also see the applied physiology. In this applied physiology, we can see the spirocytosis. What is called spirocytosis? Spirocytosis is nothing but it is a genetic disease condition in which the spectrin is produced very less. Because of this, the RBC becomes fragile. Now we can expect a question: what is the importance of spectrin. What is the importance of the spectrin in the production of RBC? See, this protein is very essential. If we lack the spectrin protein, the RBC shape will be very fragile and the stiffness of RBC will be losing. And coming to the formation we can see pluripotent cells. What is called as pluripotent cells? Pluripotent cells are also called as old cells, are also called as old cells, because these are the base for production of all type of cells. Pluripotent stem cells means they can able to produce all type of cells. And after this we can see the colony forming units, which are called as CFU, colony forming units. From this we can see proerythroblast. Proerythroblast means because before the formation of erythroblast we can see proerythroblast, and after this we can see normoblast cells. All these are the sequence of the cells. First we can see pluripotent stem cells, next colony formation units, next proerythroblast, next normoblast. After this we can see reticulocyte, and after this the erythrocyte is formed, which is also called as RBC. And coming to the specific gravity, the RBC have the specific gravity of 1.09 to 1.101. This is the specific gravity of the RBC. And factors influencing erythropoiesis. What is erythropoiesis? We told the production of RBC. And what are the factors which are influencing the erythropoiesis? We can see 85% of the erythropoietin. What is this erythropoietin? Erythropoietin is a glycoprotein. This is a glycoprotein hormone secreted by the kidneys. 85% is secreted by the kidneys and remaining 15% is secreted by the liver. So indirectly, if kidney fails, the production of erythropoietin fails, which results in the decrease in the production of RBC. So kidneys are directly involved in the production of RBCs because of production of erythropoietin. Erythropoietin is the main factor in the RBC, and because of this erythropoietin, the production of RBC will be increased. And conditions like anemia. What is anemia? Decrease in RBC or decrease in the blood count, chronic lung disease and hemorrhage, and high altitude. Chronic lung disease, high altitudes. If we go to higher altitudes like hill stations, as well as in hemorrhage condition also, this erythropoiesis will be affected. Mainly in the renal failure condition also this will be affected. Next we can see the hemopoietic growth factors. We can see IL-1, IL-2, IL-3. What are these? These are the interleukins, the factors which are used for the homeopathic growth factors. Next, coming to the maturation factors. What are the maturation factors? In this we can see maturation factors. The maturation of RBC will be done with the presence of vitamin B12. If vitamin B12 is less, you can see, if vitamin B12 is less, the maturation of RBC does not take place, which leads to a condition called megaloblastic anemia. The condition is megaloblastic anemia, means the decrease of the vitamin B12, which leads to a decrease in the production of RBC, is called megaloblastic anemia. Apart from this, the other factors like vitamin C and vitamin B, B6, thiamine and nicotinic acid are also responsible for the production of the erythropoietin. And lastly we can see the hormones. Without the effect of hormones, without the effect of the endocrine system, RBC cannot be produced. Hormones have a major role in the production of the erythropoietin. And now we can see the fate of RBC. RBC after 120 days, where it will go? After 120 days they will be destroyed, and the spleen will be taking care of the dead RBCs, which is called as graveyard of RBC. Graveyard means graveyard of RBC. See the splitting of RBC we can see in a sequential manner. First we can see the functions of RBC. What is the functions of RBC? They are used for transportation as described by the blood. They are used for the transportation of oxygen, carbon dioxide, buffering. Buffering means maintaining of the acid base balance, and they are useful for the blood group determination. This is very important point. Blood group determination can be done with the help of RBC. And next we can see in applied physiology we can see microcytosis. What is called microcytosis? Small size of RBC are called as microcytosis, or macrocytosis means big size of RBC is called as macrocytosis, and anisocytosis which refers to the irregular or different sizes of RBC are called as anisocytosis. And we can see even some variations in RBCs also. We can see sickle cell of the RBC. In this condition we can see both ends will be pointed like a sickle shape. Next we can see spirocytes. This is also one of the abnormality of the RBC. And even we can see the normal RBC are pink in color, but in these spirocytes we can see central palar, but the central palar will be absent in the spirocytes. Normal RBC contains red color and central white color, but in spirocytes these white color will be absent in spirocytes. And even we can see the target cells. What is the target cells? Target cells refers to the condition in which the accumulation of hemoglobin in center producing a bullseye or target lesions. The RBC will be allowed to form a target cells like this, which is called as target cells. Next, schizocytes. Schizocytes are the schizocytes. SCHIZOCYTES. In this condition, we can see irregular fragmentation of cells. Irregular fragmentation of cells are called as schizocytes. And coming to the last one, basophil Burr cells. The Burr cells means small irregular projections. Small irregular projections on the RBC are called as Burr cells. And coming to the application, in this we can see, as we have already discussed, the RBC will be decreased in shape and they will be having abnormality if we go to higher altitudes. Next, coming to the hemoglobin. What is hemoglobin? Hemoglobin, as discussed earlier, it is a pigment, red pigment, or the pigment present in the blood. Because of this pigment, blood is red in color. And the normal hemoglobin level in human being, that is in males, is it's 14 to 18 grams per 100 ml, and in females it is 12 to 16 grams per 100 ml. This is the normal condition, because in females it is less because of the regular menstrual flow. The hemoglobin will be very less, and in infants we can see it is 18 to 23, because they have more hemoglobin, because they RBC having very active, and these RBC have large amount of affinity to collect the oxygen. Next we can see the measurement of hemoglobin. How can we measure the hemoglobin? With the help of Sahelis, SAHLIS, with the help of Sahelis hemoglobinometry. With the help of Sahelis hemoglobinometry, we can able to measure the hemoglobin. And even this lot of procedures are used for the measuring of hemoglobin. In this we use the hematin. The blood is converted into acid hematin by using N10. This is the acid which is used for the converting of blood to hematin, which is brown in color. And after that we will distill. We will now the acid hematin is gradually diluted. This is diluted with the help of distill water, and after some time we can see the coloring, and these colors will be matched with the standardized color, and based on that the value of hemoglobin will be declared. This is called Sahel's hemoglobinometry.